In 2018, a Chinese scientist named He Jiankui shocked the world. He announced that he had edited the genes of 2 human embryos, twin girls, using CRISPR technology. He deleted a gene that makes cells vulnerable to HIV, hoping to make them immune. The twins were born healthy, and humanity crossed a line we can never uncross. For the first time, we didn't just select which genes to pass on through reproduction. We edited the code directly. We changed the human genome. And those changes are heritable, passed to future generations. He Jiankui was condemned, imprisoned, and banned from research. But the technology didn't disappear. It's faster, cheaper, and more accessible than ever. CRISPR is rewriting biology, and whether we're ready or not, it's rewriting humanity too. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. It's a mouthful, but what it does is simple. It cuts DNA at precise locations. Think of it as molecular scissors. You can delete a gene, insert a new one, or edit a single letter in the genetic code. It's faster, cheaper, and more accurate than any previous gene editing technology. And it works in almost any organism: bacteria, plants, animals, humans. CRISPR is not science fiction. It's in labs all over the world right now. Researchers are using it to cure diseases, engineer crops, resurrect extinct species, and yes, edit human embryos. The technology comes from bacteria. Bacteria have immune systems, just like we do. When a virus attacks, bacteria capture a piece of the virus's DNA and store it in their genome as a memory. If the virus attacks again, the bacteria recognize it and use CRISPR to cut up the viral DNA, destroying the infection. Scientists Jennifer Doudna and Emmanuel Charpentier realized this system could be reprogrammed. Instead of cutting viral DNA, you could cut any DNA sequence you choose. In 2012, they published the method. It exploded. Within a year, labs worldwide were using CRISPR. In 2020, Doudna and Charpentier won the Nobel Prize. CRISPR was the fastest technology to go from discovery to Nobel in modern history. The implications are staggering. Let's start with medicine. Thousands of diseases are caused by genetic mutations: sickle cell anemia, cystic fibrosis, Huntington's, muscular dystrophy. These are single-gene disorders. One mistake in the DNA, and a person suffers for life. CRISPR can fix that mistake. In 2019, a woman with sickle cell disease received CRISPR-edited cells. Her symptoms disappeared. She was functionally cured. Trials are underway for dozens of other genetic diseases. Some patients are already walking away symptom-free. This is not future medicine. It's happening now. But curing disease in living patients is different from editing embryos. When you edit an adult's cells, those changes don't pass to their children. It's somatic editing, affecting only the individual. When you edit an embryo, those changes become part of the germline. Every cell in the resulting person carries the edit. And if they have children, those children inherit the edit too. This is heritable genetic modification, and it's profoundly different. You're not just treating a patient, you're changing the human gene pool. He Jiankui knew this. He chose to edit embryos anyway. His goal was to delete the CCR5 gene, which codes for a protein HIV uses to enter cells. People with natural CCR5 mutations are resistant to HIV. He wanted to give that resistance to the twins, but the edit was imperfect. One twin had both copies of CCR5 deleted. The other had only one copy edited, and even that was mosaic. Some cells had the edit, some didn't. Worse, we don't fully understand what CCR5 does beyond HIV entry. Some studies suggest it's involved in immune response, brain function, and recovery from stroke. Deleting it might have unintended consequences. The twins are healthy now, but they're young. We won't know the long-term effects for decades. The scientific community reacted with outrage. He violated ethical norms. He didn't get proper consent. He didn't follow safety protocols. He conducted the experiment in secret and announced it as a fait accompli. He was sentenced to 3 years in prison. But the damage, or depending on your perspective, the breakthrough, was done. The first genetically edited humans exist, and they're just the beginning. Because CRISPR is not going away. It's too powerful, too cheap, too easy. A basic CRISPR kit costs a few $100s. High school students use it in biology classes. Biohackers experiment with it in garages. Pharmaceutical companies are racing to develop CRISPR therapies. Agricultural companies are editing crops to resist drought, pests, and disease. The genie is out of the bottle. The question is not whether we'll use CRISPR. The question is how and who decides. Let's talk about enhancement. So far, most CRISPR use focuses on correcting defects, curing disease, fixing mutations. But the same technology can enhance. Want stronger muscles? Edit the myostatin gene. Athletes with natural myostatin mutations have twice the muscle mass of normal people. Want higher intelligence? Edit genes associated with brain development and synaptic plasticity. Want better vision, faster reflexes, resistance to aging? All of these are theoretically possible. We already know some of the genes involved. CRISPR could, in principle, turn humans into enhanced versions of themselves. This is where things get ethically murky. Most people agree that curing disease is good. But enhancement? That depends. If everyone has access, maybe it's fine. But if only the wealthy can afford genetic enhancements, we create a genetic aristocracy, a biological upper class with inherent advantages. The gap between rich and poor becomes not just economic, but biological. Your genes determine your opportunities. And unlike wealth, which can be redistributed, genes are passed to your children. The inequality becomes hereditary, permanent. Some argue this is already happening. Wealthier parents can afford better nutrition, healthcare, and education, which indirectly affects their children's biology and opportunities. Genetic enhancement just makes it explicit. Others argue there's a moral difference. Natural inequality is unfair, but not unjust. Engineered inequality is both. If you deliberately create people with better genes, you're playing God. You're deciding who gets to be smart, strong, healthy, and who doesn't. Then there's the question of consent. When you edit an embryo, the resulting person never consented to the changes. They're stuck with whatever edits their parents chose. What if they don't want enhanced muscles? What if the edit has side effects? What if society's values change and the enhancement becomes a liability? You can't undo a germline edit. It's in every cell. It's permanent. Some bioethicists argue this violates autonomy. Others counter that parents already make irreversible decisions for children, education, religion, upbringing. Genes are just one more. There's also the problem of unintended consequences. Genes are not simple switches. They interact in complex networks. Editing one gene can affect dozens of others. The CCR5 deletion. He Jiankui made might protect against HIV, but it might also make the twins more vulnerable to West Nile virus or flu. Some studies suggest it impairs cognitive function. We don't know. The human genome is not a computer program where you change one line of code and everything else stays the same. It's an ecosystem. Changing one element ripples through the whole system. And with germline editing, those ripples propagate across generations. Evolution is another concern. For billions of years, natural selection shaped the human genome. Traits that helped survival and reproduction spread. Harmful traits were weeded out. This process is slow but effective. Now, we're bypassing it. We're choosing which genes to keep and which to discard based on our current preferences. But evolution is smarter than we are. It's tested genes in countless environments over millions of years. We're making decisions in a few decades, in controlled labs, based on incomplete knowledge. What if we edit out a gene that seems useless, but turns out to be critical in a future environment? What if we accidentally reduce genetic diversity, making humanity more vulnerable to new diseases? Some scientists argue we should proceed cautiously, edit only to cure serious diseases, avoid enhancements, ban germline editing until we understand it better. Others argue caution is a luxury we can't afford. Millions suffer from genetic diseases right now. Every year we delay is a year of preventable suffering. And if we don't pursue genetic enhancement, other countries will. China, Russia, and others have fewer ethical restrictions. If they create genetically enhanced populations, we'll fall behind. This is the slippery slope to a genetic arms race. Agriculture is another frontier. CRISPR is already used to create crops with higher yields, better nutrition, and resistance to pests and climate change. Golden rice, engineered to produce vitamin A, could prevent blindness in millions of children. CRISPR tomatoes with longer shelf life reduce food waste. Drought-resistant wheat could feed billions as climate change worsens. These aren't hypothetical. They're in trials or already on the market. Critics worry about unintended ecological effects, engineered crops outcompeting wild plants, reducing biodiversity, creating superweeds. But proponents argue we've been genetically modifying crops for 10,000 years through selective breeding. CRISPR is just faster and more precise. De-extinction is another possibility. Scientists are using CRISPR to edit elephant DNA, inserting genes from frozen mammoth DNA, with the goal of creating a mammoth-elephant hybrid. The idea is to resurrect extinct species, or at least something close. Passenger pigeons, Tasmanian tigers, even Neanderthals are theoretically possible. But should we? Extinct species went extinct for a reason, usually because their environment changed. Bringing them back without restoring their habitat is pointless or cruel. And resources spent on de-extinction could be used to save currently endangered species. Some see it as hubris. Others see it as ecological restoration. Then there's the military angle. Governments are interested in CRISPR for obvious reasons. Imagine soldiers with enhanced strength, endurance, and pain tolerance, or edited to resist biological weapons, or engineered pathogens designed to target specific populations based on their genetics. This is biowarfare 2.0. International treaties ban biological weapons, but they were written before CRISPR. The technology is advancing faster than law, and unlike nuclear weapons, which require rare materials and massive infrastructure, CRISPR works in a small lab with basic equipment. Verification and enforcement are nearly impossible. What about regulation? In most countries, germline editing in humans is banned or heavily restricted, but enforcement varies. He Jiankui operated in China, where regulations exist, but oversight was lax. After his scandal, China tightened rules, but other countries have looser standards, and even where laws exist, they're national, not global. A scientist could move to a permissive jurisdiction and continue research. Medical tourism already exists for procedures banned in some countries. Genetic enhancement tourism could follow. Wealthy people travel to get embryos edited in countries with fewer restrictions. The result is a patchwork of laws and a race to the bottom. Some propose international agreements, like the Geneva Conventions for Genetic Editing, global standards enforced by international bodies, with penalties for violations. But getting all countries to agree is difficult, and even if they do, enforcement is hard. Bioethics is culturally specific. What's considered acceptable in one country might be taboo in another. Western bioethics emphasizes individual autonomy. Other cultures prioritize collective good or family honor. Consensus is elusive. Despite the risks, CRISPR offers incredible promise. Curing genetic diseases, ending hunger, restoring ecosystems, extending lifespan, enhancing human potential. These aren't fantasies. They're realistic goals within decades. The question is whether we can pursue them responsibly. Can we use CRISPR to heal without creating inequality? Can we enhance without losing humanity? Can we move fast without breaking things? Jennifer Doudna, co-inventor of CRISPR, has said she sometimes wishes she'd never discovered it. She worries about misuse, but she also believes the benefits outweigh the risks if we proceed carefully. She advocates for public engagement, transparent research, and robust regulation. She wants society, not just scientists, to decide how CRISPR is used, because this technology affects everyone, every human, every future human. This is not a decision for researchers alone. It's a decision for humanity. So, where do we go from here? CRISPR will continue to advance. Therapies will cure more diseases. Enhancements will become possible. Germline editing will happen again, legally or otherwise. The question is not if, but how. Do we ban it and drive it underground, or regulate it and keep it in the open? Do we allow enhancements or restrict CRISPR to medical use? Do we prioritize equity or innovation? These are not scientific questions. They're moral, political, and philosophical, and they require answers soon. Because the technology is moving faster than our ethics. The future of humanity is being written, literally, in the edits we make today. And we'd better make them wisely.